Let's see now the nephrons. And the nephrons, as I mentioned earlier, these are the ones responsible for urine formation. So these are the functional units of the kidneys. And we have two classes of nephrons. We have cortical nephrons and juxtamedullary nephrons. Cortical nephrons, their name is coming because they are located in the cortex. So these are the cortical nephrons. And they are 85% of the total nephrons in our kidneys. So most of the nephrons in our kidneys are cortical nephrons. Whereas the juxtamedullary nephrons, let's understand the name. Juxta, it means near. So these are the nephrons near the medulla. These are the nephrons near the medulla. Why is that? Because they are located so down in the cortex that they are close to the medulla. So this is the medulla and this is the cortex, the two areas of the kidneys. So these nephrons are located there, and these are around like 15% of the total nephrons. Now still they are in the cortex, but because of the nephron loop, it's so long that will extend down in the medulla. And these usually are used more in dehydration conditions. So these would produce concentrated urine. Okay, so these are the two types of nephrons. However, structure-wise, both nephrons have the same components or have the same structure. We have the renal corpuscle, and the renal corpuscle is a mix of capillaries surrounded by something called the glomerular capsule or Bowman's capsule. We will understand what is that. So this is the renal corpuscle, this is the renal corpuscle in both of them. And this is the site of filtration. This is the site of filtration. Then from the corpuscle, from the renal corpuscle, we have something called the proximal convoluted tubule. Proximal because it's close to the corpuscle, and convoluted because it wraps around itself. So here, this is the proximal convoluted tubule, or the BCT. This is the BCT in the cortical nephrons. Then we will have the nephron loop. This is loop because it goes down and comes up, goes down and comes up. So it has descending and ascending branches or loops, and it will lead to something called distal convoluted tubule. Distal because now it's far from the corpuscle. It's far from the corpuscle. And you need to think of this as if the corpuscle is stretched, we will have the proximal first, then the nephron loop, then we will have the distal. So that's why this is distal and this is proximal. So this is the DCT, the DCT will lead to something called collecting ducts. These are the collecting ducts. And as you can imagine, the urine from several nephrons would drain into this area, into the collecting ducts. If we want to make some comparison between the cortical and here the juxtamedullary, we said this is 85%, this is 15%, this is percentage or number, location is cortex. Here we can say lower cortex or near medulla, hence the name. Okay. Function wise, this is normal urine. Here it's more of concentrated urine, so in case of dehydration. And also the last difference is the blood capillaries associated with it. Blood capillaries around each one. For the cortical nephrons, the blood capillaries associated or around the cortical nephron is something called peritubular capillaries. And the peritubular capillaries are mainly around the DCT and the BCT. The DCT and the BCT. There are some on the nephron loop, but not as extensive on the DCT and BCT. Whereas with the juxtamedullary nephron, the main blood supplies or the main blood capillaries are around the nephron loop, and that's called vasa recta. So this is another difference between the cortical nephrons and the juxtamedullary. Here we have vasa recta, whereas here we have the peritubular capillaries. Let's try to understand the nephron parts. And for the nephron parts, we said the first part is the renal corpuscle. And for the renal corpuscle, we said it's made out of two things. It's made out of the Bowman's capsule. This is the Bowman capsule. You can think of it as cells that surrounding something. So there is creating a ball, okay? And within this ball, there is the glomerulus. So the Bowman capsule is just layers of cells. But we will focus more on is the glomerulus. Now the glomerulus is basically blood capillaries that enters the Bowman's capsule, it wraps around itself and then exits. So this is the glomerulus. And as you can see here, it enters and exits from the Bowman's capsule. Why this is happening? Because this is the site of filtration. Okay, remember, this is the site of filtration. So how this filtration is happening? Before going there, we need to go back on the slide that shows us the blood supplies to the kidney. If you remember, we said the cortical radiating artery, then we will have the nephron. Now in the nephron, that would lead the cortical radiating artery, to something called afferent arteriole. Afferent arteriole, which will lead to the glomerulus. And from the glomerulus, we will have the efferent arteriole. Remember, we said the Bowman's capsule, blood comes in to the glomerulus and blood comes out. So this is the afferent arteriole and this is the efferent arteriole. And here is the glomerulus. Now from there, from the efferent arteriole, it will go either to the pretubular capillaries or the vasa recta, depending on the nephron itself. And then from there, it will be drained to the cortical radiate vein. Okay, so now we understand that the blood coming to the glomerulus through something called afferent arteriole and exit from the efferent arteriole. Now the afferent is a little bit bigger than the efferent. So there is more blood coming, less blood exiting. Okay, and this is critical because we need to maintain certain blood pressure in the glomerulus, which we will understand later. But at least that's the concept. That's the glomerulus surrounded by Bowman's capsule. We said for the renal corpuscle, its main function is filtration. So here within the glomerulus, we will have a filtration membrane. Now for the renal corpuscle in general, we can think of it as one structure. So we have the Bowman's capsule. This will serve as the parietal layer of the corpuscle, whereas there is a visceral layer. And this visceral layer or the visceral layer is made out of special cells. These are called podocytes. This is a podocyte. This cell is a podocyte. This cell is a podocyte and podocyte. So these blood capillaries in the glomerulus are surrounded or covered by these podocytes. Podocytes, it means cells with legs or extensions. So as you can see, this is the cell and these are its extension, and they go really tiny, really deep to cover most of these blood capillaries. These extensions, we are called them pedicles. So what we are looking at here are the pedicles of the podocytes. That would create a filtration membrane. Now we need to remember that the main function of the glomerulus is filtration. So filtration is happening through a filtration membrane. So we will have membrane. And this membrane is made out of 3 layers. The first layer is the openings, or here they are called them the fenestrations or the windows in the blood capillaries, in the glomerular capillary. So here we are talking about these blood capillaries. See these dots here? So these, you can think of them as windows within the blood capillary. That would allow certain materials that are less than 8 nanometer, this is 1/1 million of a meter, to pass through this window. So the first layer of the filtration membrane is the capillary endothelium, these windows or the fenestrations or the pores. Then we will have the basement membrane that would attach the blood capillaries to the podocytes. That's the basement membrane. Then the third layer is the food process of the podocyte or the pedicles. Each one of these is a pedicle. Since these pedicles, if we go back here, since these pedicles are not completely covering the blood capillary, so there is some space. From this space, a filtrate is passing. So from this space, a filtrate is passing. And since here we are talking about 8 nanometer, so we are expecting here it's the same size. Now 8 nanometer, it means most of the proteins, or actually all of the proteins, all of amino acids, all of course all of the cells will be denied, will be blocked. They cannot exit from here because it's super small. And that is the concept of filtration. When you are, for example, brewing coffee, you will put a filter and you will put the ground coffee, right? What goes through the filter is just the water, whereas the grounds of the coffee itself would remain within the filter. So that's the same concept here. These big structures would remain in the plasma. What passes by is mainly water-carrying solutes. Water-carrying solutes. And this solutes has to be, of course, less than nanometers. So we're talking about ions. Sometimes we'll see some glucose, sometimes pass some individual amino acids. But these, under certain conditions that they will pass and most likely will be reabsorbed. So I will stop here, and in the following video we'll continue talking about the parts of the nephron.